Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Bioscience with Shatrin Savati. And today I'm going to discuss about the PV interacting RNA biogenesis. In my previous lecture, I have discussed in detail about the introduction, discovery, PV interacting RNA complexes, and many more things in my previous. If you are taking interest in research and about the non-coding RNA, about the silencing mechanism, so must watch this lecture till the end, and all the lectures related to the biogenesis and PV interacting RNA mechanism of action. So must watch all the lectures and kindly share with others. And if you did not subscribe to my channel, kindly do subscribe. Okay, now I'm starting my lecture. PV interacting RNA in animal gonads, small non-coding RNA, as I have discussed, that termed PV interacting RNA mediate TE silencing to ensure genome integrity during germ cell development. Actually, piRNA not only you can say that the help in the spermatogenesis and means the growth and development of the sperm, but also help in the development of the gonads too. And how do they work? They do the silencing of the TE. TE means transposable elements or transposons. They do all this because of the silencing of the transposons. So piRNA clusters are genetic elements that regulate the activity of the TE. And if you need all the articles, I will provide you the link of all these articles which I use for creating this lecture. Okay, so must read all these articles, very interesting, and they give you knowledge related to the piRNA. piRNA clusters are transcribed as long non-coding RNAs. And you know that this is 24 to 32 nucleotide in length. And this piRNA is synthesized by the RNA polymerase 2. You know that there are 3 types of the RNA polymerase, 1, 2, and 3. And this RNA polymerase 2 help in the formation of messenger RNA 2. And here it work in the formation of the piRNA, or in the transcription of the piRNA. So despite this fact, the chromatin of the piRNA cluster is enriched in the histone 3 lysine 9 trimethylation, H3K9Me3. This is abbreviated as H3K9Me3. This is histone 3 lysine 9 trimethylation, which is usually found on silent. Must be in this histone 3 lysine 9K trimethylation is usually found on silent, means this is heterochromatic region, and is thought to be a mark that suppresses transcription. It is thought to be a mark that is responsible for the suppression of the transcription. But here it helps in the silencing of the transcription, and how it will do, I will show you. And something very interesting I want to share, that the biogenesis of actually the piRNA is still under consideration and not fully understood. Research is going on, and I will share step by step. Okay, surprisingly, the presence of this He3K9Me3 mark does not interfere with the piRNA precursor transcription. Means it do not interfere with the piRNA transcription, but is in fact required piRNA expression. Is it clear? So piRNA transcription is possible because of this H3K9Me3, which is the mark which is mostly on silent in, or you can say that in the heterochromatin. This is the heterochromatin region, and this help in the formation, or you can say that in the transcription of the piRNA. Okay. Now, as I'm discussing all these important information about the piRNA, and this is all about the Drosophila and the later on in the mouse. So I'm discussing all these things related to the Drosophila and mouse, and now in the human being, the research is going on about the role of the piRNA. So 3 piRNA are responsible for gonad development and transposon silencing. Must keep it in your mind. These piRNA is actually responsible for the silencing of the transposon and also help in the gonad development, and you can say that the gametogenesis. So first one is the AGO3, and I have discussed in my previous lecture AGO3 argonate protein family, only required for female fertility and cleave transposon transcript in the cytoplasm. Second one is the AUBERGINE, required for both male and female fertility and regulate target transposons at the transcription level. And the third one is the PV, the proteins, required for both male and female fertility, target transposons at the transcription level. Okay, now I am sharing a very important thing here. I'm just underlining all these points. Here you can see this help. This first one, AGO3, cleave transposon at transcript in the cytoplasm. So it do the silencing in the cytoplasm. Second one cause the cleavage, which is AUBERGINE, responsible for the silencing at the transcription level. You can see here, this is at the transcription level. And the third one, PV, also help in the targeting of the transposons at the transcription level. Is it clear? So AUBERGINEs and PV target the transposon at the transcription, meaning the nucleus, and AGO3 in the cytoplasm, which I will show you later on how it work. Okay, now I'm moving towards the next slide. The sources of piRNA production in the genome. There are 3 sources which I will show you. Number 1 is the single-stranded piRNA cluster. I'm showing you here. You can see this is the DNA, and in that DNA you can see this is the single-stranded first one. I am showing you this first one. This is the single-stranded piRNA cluster, and the second one is the Genic piRNA, and this Genic piRNA is present on the 3 UTR. UTR means the untranslated region, and the third one is the dual-stranded piRNA cluster. So this single-stranded piRNA just synthesized from the one strand of the DNA. This Genic, which is Genic, why it is called Genic? Because it is present close to that gene, to which it later on do the silencing that transposons. Okay, and the third one, dual stranded, from both the strands of the DNA. It will that both dual strands synthesize the piRNA. In this case, just one. In this case, you can say the Genic, and this is the dual. Is it clear? Okay, now I'm moving back to show you. Okay, now you can see here the single-stranded piRNA cluster, Flam locus in the Drosophila, 180 kb in length. Genic piRNA cluster, 3 UTR of coding gene. The coding gene which that piRNA will regulate. Is it clear? So this is present at the 3 UTR, untranslated region of that coding gene. And the dual-strand piRNA cluster, this is 42 AB cluster in the germline cells in the Drosophila. So this is very important to understand the concept and you can say that the position, or you can say that the source of piRNA production in the genome. I hope so. It will be clear upon you people. Okay, now I will start the biogenesis of piRNA, which I will discuss in my next lecture. There are two major pathways generate piRNA. Number one is primary processing pathway, and the number two is the ping pong cycle that amplifies secondary piRNA operate in the germline cells. Is it clear? So how many pathways? And these pathways are still under consideration and not yet, you can say that, fully understood. So two major paths. First one is the primary and the second one is the ping pong, which I will discuss in my next lecture. So must watch, must share, and do subscribe the Bioscience with Chatri in Sawati. I will be back. Till then, Allah Hafiz.